Welcome back to Jalen Bio. Today's module is on the molecular transport. Transport of molecules along the concentration gradients. Molecules move randomly in a process called Brownian motion. One important function of the plasma membrane is to regulate materials that get to go in and out of the cell. Membrane transport can be either classified as passive or active depending on the cellular energy expended. Passive processes require no additional energy other than the kinetic energy already present during that net movement of one concentration gradient to another. Some examples are like diffusion. And diffusion continues until the substance concentration on each side reaches an equilibrium. Equilibrium is established once a substance reaches the equal distribution in an area. So you can see on the picture that when it goes to the right side, this is when it achieves equilibrium. These little particles are said to move down the gradient because it's going from an area of higher down to an area of lower over here. Active transport, on the other side, requires energy in the form of ATP to move solids against the concentration gradient across the membrane. Some examples of this include primary active transport, which we'll talk about in the next couple slides. Solids vary in their membrane permeability. What are permeable? These are generally going to be items that are small, nonpolar solutes, oxygen, carbon dioxide, steroid hormones. These ones will go through the membrane without any problem. Impermeable solutes include anything that is large and polar, as well as the macromolecules such as carbohydrates, proteins, and anything with a charge. Passive transport number 1, simple diffusion. Simple diffusion is a direct movement of solids across a semi-permeable membrane. Simple diffusion is where small nonpolar molecules squeeze between phospholipid molecules of the plasma membrane from an area of higher concentration to an area of lower concentration. This plasma membrane cannot regulate simple diffusion. It is solely based on the concentration gradient. So, as a result, anything that is small, charged ions, or polar solutes are unassisted by the plasma membrane will be blocked from entering the cell, where anything that is nonpolar, steroid, gas-based can go right through. The rate of diffusion depends on the steepness of the concentration gradient or the temperature. Steeper the gradient, the higher and faster the rate of diffusion. The higher the temperature equals to higher kinetic energy, and therefore accelerates the rate of diffusion. Here are some key points. Solids diffuse from one side with the higher to the other side that's lower. Solids move independently. After equilibrium is achieved, solids will not move anymore. And this is only going to be limited to short distances. As you go from the left picture to the right picture, it has achieved equilibrium. On the bottom picture, you can see two sides. Again, both of these solids, both the red and blue solids, are able to go through the membrane because of these small little openings. It then also achieves equilibrium. Passive transport number 2, osmosis. The plasma membrane is semi-permeable, and it only allows certain solids to pass through and is barrier to others. This membrane is also selectively permeable because it regulates the movement of solids. Osmosis is a passive process by which water moves across the plasma membrane in an effort to equalize its concentration on both sides. This is the movement of water across the membrane due to the imbalances of the solute concentration. Aquaporins are protein channels that allow water to move across a selectively permeable membrane. Solids are then classified either as permeable or non-permeable. The permeable solids can pass through the membrane, whereas non-permeable solids cannot. As a result of this movement, there's going to be osmotic pressure. Osmotic pressure is generated from the amount of water moving across the plasma membrane via osmosis. So it is the pushing in of the fluid to one side. On the other side, we have hydrostatic pressure, which is where a fluid wields a certain amount of pressure upon the internal walls of the container. So this is the force that resists it. As a result, we can describe tonicity, and this is where the cell's volume and pressure changes based on osmosis. There are 3 types. The first type is called isotonic. This exists when the amount of water and solids on both sides, inside and outside, are relatively equal. We call this isotonic. Hypotonic exists when the cytosol has a higher concentration of solids than the interstitial fluid. This will then cause the water to enter the cell in an effort to equalize the solid water concentration. So there's more stuff inside the cell. If too much water enters the cell, it could risk lysis and rupture. And in a hypertonic solution, there's more stuff on the interstitial fluid than on the cytosol, therefore causing water to exit the cell. Facilitated diffusion. Facilitated diffusion is when there's a movement of impermeable solids using a transport protein. 2 types of facilitated diffusion exist. There's a channel-mediated diffusion and a carrier-mediated diffusion. The channel-mediated diffusion is where a certain ion can move across the membrane through a water-filled ion-specific channel, like right here. This is a specific channel that moves the ion from one side to the other side. Sometimes it could be leak channels that remain open at all times, and sometimes it could be closed. Another type of channel is called a carrier protein. The carrier-mediated diffusion occurs when a small polar molecule, such as a simple sugar or amino acid, is assisted by a carrier protein and allows to move it from one side to the other side. A carrier that transports one substance is called a uniporter. Uni means one, porter means one direction, so it's a uniporter. The number of channels in carriers determine what we call the maximal rate of transport, which is referred to as transport maximum. This refers to the maximal amount of speed by which a substance can be moved into a cell. A cell can alter the transport rate by changing the number of channels or carriers to meet the needs of that cell. Here is a chart showing you the types of channel proteins. Voltage-gated channels, which are responded by changes in charges and ions. Ligand-gated channels, which can respond to a binding of a small chemical compound or messenger or hormone. Ligand-gated channels, which ligand and signal is found on the cytosol side, inside the cell. And we also have mechanical sensitive receptors that can change and open based on stretching and expansion of the cell membrane. Active transport. Active transport requires energy from ATP to move solids against the concentration gradient across the plasma membrane. Primary active transport takes a phosphate from ATP and adds it to a transport protein in order to change the protein's shape. This will then result in a change that allows the solute to cross the membrane. We have a lot of these cell-lipid pumps called ion pumps, which are active transport proteins that help in the assisting of movement of ions across the membrane. One example is called the sodium-potassium pump. This is an ion pump where potassium is moved into the cell against its gradient, while sodium is moved out of the cell against its gradient. Other types of pumps include calcium pumps and hydrogen pumps. Let's take a look. So 3 sodium ions are loaded into the binding site. ATP is used. This allows a change in shape of the protein. Then the 2 sodiums are then released out of the cell to the outside against the gradient, allowing 2 potassium, shown here in purple, to bind onto the binding site. And as the pyrophosphate is released, it will close the external side, allowing the potassium to enter into the cell. It transfers a 3 sodium to 2 potassium ratio. The proton pump is an example of a channel protein that performs active transport. ATP is used to phosphorylate the pump. ADP plus pyrophosphate becomes ATP, and that allows the opening of the channel, permitting the flow of protons to the outside against its gradient. Co-transporters use ATP indirectly to perform active transport. Co-transporters are also known as secondary active transport, since it is connected to a primary active pump. The kinetic energy moving down one substance, usually sodium, down its gradient, provides the power to pump another substance against its gradient. There are 2 types of secondary active transport. There's a symporter and there is an antiporter. Symport is a secondary active transport that occurs when they move the 2 substances in the same direction. Antiport is when you have a secondary active transport that moves 2 substances in the opposite direction. So in this case, you can see that the sodium-potassium pump is working to do its job by moving sodiums out to the cell and potassiums into the cell. And then the sodium on the outside can co-transport with another symporter to allow glucose to come in, and then sodium goes back into the cell. So 2 pumps work together to move glucose back into the cell.